the founder of the company is Dr. Frost, and she is a PhD chemist, University of Michigan. And when she was at the University of Michigan, she was working with materials that would release nitric oxide. Nitric oxide is very different from nitrous oxide. Nitrous oxide is what you get at the dentist's office and they take home over the weekend. The nitric oxide is a free radical gas that is very ubiquitous. You find it in every living cell, and it is generated in quantity in other places in your body, but for certain in your sinuses. I'm battling a cold, and so as you speak and as you breathe nitric oxide gas that's generated in your sinuses into your lungs, and it helps protect your lungs from bacteria and viruses, molds, those kinds of things. So the gas is natural. It is not particularly easy to handle, or it wasn't. Pardon me. And then Dr. Frost came up with a way to generate in a polymer chemical reaction that releases the gas.
So I'm a very visual person. I don't need a slide deck, but can you show us anything so I can get my head around what we're talking about?
Yeah. I have a slide deck, which I was going to use, but the product essentially is, think of a Ziploc bag, and the inside of the Ziploc bag is coated with the polymer that Dr. Frost created.
Okay.
You take a medical device and place it in the Ziploc bag.
Yes. You
seal it, and you ship the product to your distribution system.
Mm-hmm.
There is no need for a sterilizing chamber. There's no need to ship it to and from a sterilizer. It's a self-sterilizing package. It's been tried before with other elements, but this one is pretty easy to use, and you don't have to off gas. You don't have to precondition. Mm-hmm. You just put whatever it is you want, sterilize, package, you seal it, and it goes.
So let me ask you, do you produce a film?
We don't produce anything because
well, maybe that's why you have no revenue.
That's one of the reasons. Yeah. We have, they're called contract manufacturers, which I've become familiar with.
Andre knows a thing or two about contract manufacturing.
And they make what we describe as the juice. And then that juice gets shipped to coater converters. They coat film with it, and then they convert it into whatever you want.
Yeah, we're getting to the point where I'm going to need to see something because I'm getting confused. Bruce, you have your hand up?
Yeah, I do. Let me ask you a question, if you don't mind. So this bag is coated, if I understand correctly, with something, maybe, is it nitrous oxide? I heard you mention that. I don't know if that's the compound. And if so, if there is a coating involved there, does it sterilize the product that's been put into the bag externally? In other words, the outside of the component or piece of the medical device that's been put inside the bag? Or does it actually, like a ETO or an autoclave or something, go into the creaks and crevices and sterilize the internal parts of the device?
It does. It is a gas sterilization system, if you will, the same as ETO. And it's actually, the polymer generates the gas, and it is, I should talk about the safety aspects, but I'd like to make sure that I've answered the questions fully as you would like. So maybe this slide will help.
Please.
Nitric.
Are you trying to put it up? Because we don't yet see it.
Oh, you're not seeing it?
No, sir.
No.
Oh, well, that's probably a bad
Joe,
thing.
how there's 2 layers in the Kleenex?
Yeah.
that's what they're doing. They're putting an extra layer on a film. So you can't really see it unless they put a colorant in it. So it's a laminate
but-
layer.
But, and he'll show us when we see the slide, which we still don't.
Yeah, I'm having trouble with that.
Here on the bottom of the screen should be a green button that says share.
I know, but it's not, for some reason, it is not showing.
You can send the slide deck to Joe and he can show it, or me.
Okay, here we go. I'll share. Here we go. And
There we go. Good. We see your email, but I'm sure we'll get there.
no,
So I imagine if you click on whatever icon, is it a PDF? Is it a PowerPoint?
it is.
There we go. There we go. We have it. So
Great.
in Jan's example, she showed us a tissue with 2 layers. I'm thinking that you make one of the layers, but you don't make anything. So maybe your next slide will help me visualize what it is how you do what you do.
I don't know if this next slide is going to, but I will show you.
Oh my.
I'm having a great time, aren't I?
No, I'm just looking at your desktop. You need an admin to clean that shit up for you.
Yeah, it has been mentioned previously. So let me describe it again. There is a polymer that is coated on top of a film. We have a company that makes the polymer, and then we use companies that coat film. So
Repeat that again. We make.
there are companies that make the polymer.
Okay.
It is shipped to companies that make pouches.
Okay.
They coat film
Mm-hmm.
with the magic juice. It gets fashioned into pouches,
Okay.
and then the pouch is used as easily as I described it.
Okay, so you have a middleman essentially as a contract manufacturer, but if it were in-house, you make a polymer. Your technology, you license the patent to make a polymer.
Yes.
Okay. That I can visualize.
Okay. And then the polymer is used to make pouches.
Uh-huh.
And it is at that point where some of the details of the sterilization might be of interest. As you started, it is a gas sterilization method. It's not a method that deposits a sterilant on the surface of a medical device. It's truly gas. And so the gas laws, I should interrupt this really quickly. There's some chemistry that I'm going to talk about and some physics. The last chemistry class I had was in 1973. So there's a possibility that I may not be the best source for all of the chemistry, but that which we will talk about today, I think I can describe.
I found chemistry exceptionally difficult as a sophomore in high school, but please continue.
My experience was similar. So what makes this attractive is there is no shipping to a company that does sterilizing. There is no chamber that you put items in and have the gas come from the outside and swirl around the packages and have it find its way into the packages. And then it has to be extracted again. It doesn't have to be extracted. It is so benign that there isn't anything that we have found so far. We've tested more than 60 medical devices so far, and we haven't caused any damage to any of them. We can sterilize items that have electronics without any issue. For example, there's a company that's working on getting packaging reworked for them. They make ablation devices, and about 20% of their ablation devices came back from their sterilization method with the buttons not working. And apparently they got some response from surgeons using them that they would get the catheter up into the patient's heart and then the button didn't work. And they didn't think that was very funny. So they needed a new sterilization method, and they have tested with ours and they're pleased with it.
Okay.
So it has this wide range
All right.
of
As your
materials.
host, may I gently insist you show me a picture of something, not words. Let me see something, because I'm struggling. More, keep going. Is that the only picture you have, a stock photo?
Pretty much.
All right, then I'll just have my hand up with questions still.
That's fine. I'm good at answering questions.
No, my questions
usually.
going to be, I'm still. Well, maybe Andre, well, who has his hand up? Thanks. As a contract manufacturer, have you dealt with anything like this?
Yeah, as a matter of fact, Joe, very heavily involved with this on two aspects. Number one, we actually helped develop a product for a client that produces nitric oxide for use in treatment of diabetic foot ulcers. It uses a plasma, though, electronic plasma, to create 1,300 parts per million of nitric oxide to be treated upon the wound. But on the other side of it, we've been involved with a number of devices that require sterilization, that exactly what you say, Kurt, that can get damaged by the different methods of sterilization in the electronic aspects. So I'm very curious, considering the half-life of nitric oxide, how does the polymer continue to maintain the level of nitric oxide with the less than a second half-life of nitric oxide?
Well, inside a completely sealed container, like a pouch,
Right.
The first thing that nitric oxide does is it scavenges all the oxygen.
Right.
Eliminates the oxygen in the package. And then as the amount of NO reaches a certain part per million, that's when it starts sterilizing.
Right.
And so even though our polymer will continue to emit NO until it gets to the steady state level, and at that point, that's how it sterilizes. And I understand the foot ulcer circumstance. And Megan has done a lot of work with similar things. And so someday, if you want to talk about that, we can go into what we do, because we can also sterilize tissue.
No, I'd love to speak to you about that. I achieved a couple of patents on developing the nitric oxide with high level plasma. And also I'm very interested on the sterilization. We actually have a product that we're working on right now for another customer that has a very complex camera, electronic camera. And so far the ETO has phased out the lens that's on that camera. It's a 1.2 millimeter camera, very tiny lens. And when it went through ETO, it affected the lens. So I'm very curious if your sterilization method would preserve the lens on this.
Most likely it will, because we have tested with other optics without any impact.
Okay.
And that's been glass. And also there's some kind of polymer that they make some optics out of, and I don't know anything about it.
Certainly like to connect with you on both aspects.
Sounds great. Be happy to do it. There are some medical devices that also have, in addition to the electronics, they have little motors. And those motors don't do well with ETO. They don't do well with chlorine dioxide or hydrogen peroxide. So those looks like those would be good candidates for us as well. And we're trying to do we're getting to doing the validation testing.
Luke?
I'm sorry.
I was calling on Luke with his hand raised.
Oh,
Yes. I have a few questions, technical questions. But also, I first like to tell you a story that in my former company, we developed vaginal probes, and the customer wanted to have them sterilized. So we had them sterilized by Etox. It was packaged and it was sent to us. And by that moment, the sterility chain was already broken. So a lot of people don't realize that if you have something packaged sterile not in your way, but in the ordinary way, and you don't have the conditions really under control until you have them at your customer, that the sterility chain is broken and it's not longer sterile. But then again, they were vaginal probes, and the things you stuff into a vagina is usually not sterile. So it's enough to have them disinfected. And for the women who get the procedure, the diagnostic procedure for measuring the pelvic muscles, it is nice to see that they get it out of an individual package and that it looked like it hasn't been touched by anything else at all. So it had its purpose, but it was by no means sterile. And I guess with your product, it would have been sterile from the moment we package it. Correct?
Correct.
Okay. But then how do you prove that it's, because sterilization starts when you start packaging your product, how do you prove it's sterile when it arrives at the customer? That's
Two
my
ways.
first question.
One is there's a parametric release that is similar to the parametric release of other systems. And secondly, we have a chemical indicator that describes, it's a dosimeter. It describes the amount of gas for the right amount of time. It changes color. And so you can be assured that way as well.
So the outside indicates, it says, don't open this package until the indicator shows green or something like that.
Right.
Ah, that's a nice system.
And
then-
it is green, by the way.
It is green. I couldn't imagine that. Next question is, for our finger cuff, I'm now really into high frequency welding of polyurethane films. And having pollution on the film makes it hard to weld them. So I imagine that your product, if you have a pouch, you first make a pouch and then you try to apply the product from the inside, something like that? Or is it weldable with your product applied to the film?
To answer your question about weldable, I don't know. I do know that
If you want to make a pouch, you usually have several films and you weld them together. You have like a air mattress. You have your welding seams on the outside, so that makes your pouch. But if you put your polymer to the product, you make your pouch off, and you then want to weld them together. Well,
Yeah, we
personally,
call
I
it
had experience-
sealing. He says you can seal right through it.
oh yeah,
you
it's
seal right
sealing.
through. That-
Yes.
okay, that's a nice term.
Yeah, we didn't figure that out. The packaging company did.
Okay, but then if you seal it together, then your package must keep the seal together or something.
Yes.
Okay. Well, that answers the question.
And
that's
then,
part of package engineering is making sure the seals are good and they stay good all through distribution.
Mhm.
And I wanted to know if he had a sachet, a little pouch he could throw into a pouch I've already validated as being acceptable. And they're still working on that technology.
Okay.
the answer is yes. We
So
have,
you have an indicator that shows that it's sealed, that it's sterile. But do you
Yes.
also have an indicator when the seal is broken on the way?
No.
there isn't one that it's broken along the way other than what everybody else's has, because nobody else has that either.
Right. That's part of the validation process that you have to do with distribution testing and in aging.
Okay.
Because nobody has that I know of. Unless you get a seal that actually changes color,
Mm-hmm.
but then you're not checking the rest of the package for holes.
No, you're
One
not.
of the things I can tell you that is unique with ours is sterilant. Let's say you get a pinhole in one of these pouches. With every other sterilization method, whatever gets introduced through that pinhole is not treated. With our system, it gets continuously treated. So you have a higher likelihood of a sterile product, even with a pinhole with ours than with any others.
I guess that's a valid argument.
We're not taking that to the FDA yet because we don't want to have to prove all that. But but I'm telling you, I mean, it is a characteristic of our product.
I'm intrigued by your last statement. If you don't want to take it to FDA and prove all that, how will you get to revenue? Because your end users or your customers, ultimate customers, won't be able to use your product.
No,
right?
no, it's only the pinhole problem that we're not taking to the FDA. Everything
So
else.
the rest of it,
for the
so
FDA.
tell us
We-
about what regulatory, have you I don't know, is this an FDA cleared technology?
Not yet. No, we've been working with them for a few years. And in my prior life working with the FDA, I didn't use the terms that I'm about to use. They've been cooperative, they've been helpful, they've been friendly. And in my prior life, I never used any of those terms when I was talking about the FDA. So it has been a whole new experience. And we don't have approval yet, but we think we are very close.
You've done a pre-sub at this point?
Oh, a bunch.
Okay. What's your source of funding at this point? Are we at seed? Are we at series? For friends and family? How much has been raised since you've been there?
Raised 9 million, and it's all been friends and family.
Okay, you have some rich friends.
They're just easily convinced, you know.
Rick asked, what is parametric release? What does that mean?
It's by the parameters that the device is made, by the parameters that it's made, or the package is made, allows you to determine that each package has been sterilized. It's not practical to put a biological indicator inside of every package, and no one does that.
So that's the alternative to parametric release is biological indicator confirmation. Are there any other things that would be, are there any other methods of confirming sterilization that this would contrast with?
No, I think we're pretty much, we're on the same spectrum, if you will, for all the other gas sterilization methods.
To me, it's more
But-
like radiation, because they do have a dosimeter for that shows that your whole thing has been sterilized. And you go through and you set up your packaging system and prove through validation methods that it works. There's actually a guidance document from the FDA with parametric release information in it too. And ISO has a lot of standards on sterilization, ISO and AMI.
No, I just wanted to make sure that I had the right idea. I'm a mechanical engineer. I also work with polymers and flexible things and all kinds of stuff in design. And that's just a term that I hadn't come across lately. So thanks.
Look at all these new
Everybody
friends you're
strives for it.
making, Kurt.
I'm sorry.
I say, look at all these new friends you're making.
Yes, I see that.
Andre?
Yeah, Kurt, just to follow through on it. So we're a contract manufacturer of electronic devices. We make something that has to be sterilized. We have to package them in bulk, ship them out to a sterilizing facility, get them sterilized and single packed, and then come back. Are you saying that with your situation, we could literally put it in preformed pouches, heat seal it here, and not have to send it out for sterilization?
That's what he's saying.
Correct.
That's major.
Oh yeah,
is
that's why I like it.
That's
We
major.
just have this issue of getting through FDA first.
Yeah, but I hope you get through right away because I'm ready to use your product.
Look at that. A
We-
pre-sale.
We thank
Tell
you.
that to your friends and family.
Yeah, there you go. So I would say this. My crystal ball for predicting when the FDA will give us approval has been almost as good as when I predicted the winner of the Super Bowl. And that isn't very good. So I don't know when we're going to get it, but it does look like it's months, couple months.
That's quick.
Well, yeah, it's quick if you're at this end of the testing regimen. It wasn't quick 3.5 years ago when we started.
Well, it's
But
quick
then
to us.
again,
I'm
he's
sure it's
predicting
long for
this
you.
with the accuracy of a gambler who loses, so. Kevin, you had something in the comments?
Yeah, I've got two questions. The first is what kind of base polymer you're using, just because that affects, well, although actually, I guess if you're selling the actual pouches, that's kind of your problem. I was going to say, if you're doing lamination, you've got, if you want to put it on something else, you got to worry about what your base polymer is.
I think Megan says that she can work with 8 different base polymers. and there's probably more. She has 42 polymers that she's created.
Yeah.
For different, she calls it substrates for the film. That's what you're talking about, I think, right?
Yep. Yep.
So far we haven't found a substrate we don't like.
Okay. And I'm just kind of curious, just on the FDA approval, kind of what that looks like for a system like this. How many different scenarios you have to validate against to actually get a claim approved?
Where are you located?
Well, we don't really have a central location for our business, but if there
Where do
was
you
one,
live?
I live in Midland, Michigan, but the test facility where Megan Frost is, and that's really the heart of the company, that's in Houghton, Michigan. I don't know if you know about Houghton, Michigan. From her back deck, we kid her that she can see the Arctic Circle. She is about as far north as you can get in the
Americas.
in the United States. And then I already lost the question that you asked about.
No, he asked a real question and then I did what I do. Sorry.
No, just kind of, what that validation involves, and how many different ways and different scenarios you need to actually validate to get a claim approved, or how narrow that claim is going to have to be in order to get validated.
I can tell you that there are 3 things in front of them: the primary sterilization in a pouch, plus the 2 ancillaries, the biological indicator and the chemical indicator.
Yeah.
Kurt, I very much would like to go on Joe H. turf, because I'm amazed he didn't ask the question yet. So what is your marketing strategy? Are you trying to sell your stuff to companies who wanted to ship stuff sterilized, or are you trying to convince a package manufacturer like say if you go to the post office, you get those standard size boxes, and you can get a box in which you can put stuff in, get it sterilized and send it away. So what is your marketing strategy in this?
I didn't ask the question yet because without FDA, he's got no marketing strategy.
Well, I have a strategy.
Upfront.
It's just not in effect yet. Yeah, the strategy goes like this. The packaging companies that will take our juice and coat the film and then turn it into various size pouches, they all have customers. And they're going to their customers as we speak and saying, this is going to be available soon. Is this of interest to you? In addition, Megan and I have tested challenge devices for 40 companies and around 70 devices over the last couple of years. So we did both. We have come to know lots, maybe 12 of the biggest 20 medical device manufacturers in the world, tested products for all of them. And I can tell you that we have always been able to prove sterility, and we've always been able to prove that we did no damage to their device. So you're right. This FDA clearance for us is kind of important.
Kevin,
We're
would
ready.
you give Kurt your background in polymers so he knows a little about you?
Yeah, so we're kind of coming at the problem from a different direction. I mean, we're not looking to sterilize. We're looking to actually protect products from contamination kind of during use. So we put an antimicrobial into a polymer. Our competition is primarily the metal-based antimicrobials, the silvers and copper, zinc compounds. Our product works. It's a hell of a lot more powerful. Most of those products work very slow with a pretty subdued kill rate. So our target is a little bit different. But for us, part of why I was asking what you're putting in, like for us, we have our own issues, on compatibility with polymers and our antimicrobial. So we're primarily in polyolefins, polypropylene, polyethylene. And for us, based on the founders' experience, we actually started with fiber-based applications. So for synthetic textiles, to start, and we're moving into molded products as well.
We're already working with molded products, and we would like to work with textiles because we think that companies that made drapes and gowns out of essentially our material embedded in your material would have a fair amount of market interest.
Yeah.
So we have worked on that already, but we're not as close to being able to introduce it.
Yeah. I mean, the challenge for us is for, some of our big applications in healthcare, things that, spread contamination in a healthcare environment aren't medical devices, and that turns it into an EPA registration, which is a hell of a lot more expensive and longer regulatory pathway to get through. And that's kind of our challenge. And, so something like, hospital curtains is actually one of the big vectors. We've had back and forth discussions with EPA, and one of the big problems there is no accepted protocol to test long-term claims for that. So that's a big expense just in itself, coming up with a protocol and validating a protocol to submit to EPA to begin with.
Well, disposable curtains.
No, we're talking about the long-term ones. So, disposable is less of an issue. It's more so the ones that they leave up there for weeks or months at a time, particularly in things like underfunded nursing homes.
We can do months.
Yeah.
I don't know about years, but I know we can do months.
Yeah.
And because the release is very close, of course, to the fabric, and then it's a 1 second half-life. It doesn't take a high dose, but the gas does keep bacteria from being able to adhere to a surface. And we spent a lot of money on testing it. So we are very effective against viruses and tuberculosis. And tuberculosis is something that hasn't been easy for some people to do.
Yeah.
So we have a division of the company. I'll just tell you about it really quick, where we sterilize tissue. So tissue, for example, corneas that are transplanted to avoid cross-contamination. There's certain things they do, but it's a disinfection process instead of a sterilization process. And we don't damage the corneas. Same thing's true with amniotic tissue. So it's just very gentle on everything except the bad bugs.
Before I give the floor to Luke, because he has a question that may be more Jan than Kurt, or both of you. There are a number of attendees on today's call whose names I don't recognize. And I wonder, given the concentration of people on the panel who are intimately familiar with what you're talking about and have shown great personal interest in what you're working on, I wonder if that's the case for those in attendance as well, these names I've not seen before. So if you're willing, folks, to just write a note in the Q&A about your background and why you joined this call in particular, because I, again, see some names I don't typically see. I think that would be interesting not only to Kurt, but to your host. Luke, you have the floor.
Okay, I had a question in the chat. Are you allowed to send a sterilized product all over the world, or are there regional regulations that may prohibit that?
I think there's
Yeah.
regional regulations. Yeah.
Yeah, there are regional ones, but there are ISO documents for sterilization that are used all over the world. And a lot of it comes through AMI, which is a US regulation. And I did check to make sure. And I know that Dr. Frost is working with AMI to develop a document for sterilization through them, which then can be submitted to the ISO community for use around the world. So that'll take time too.
Yeah, but
It-
if it's an ISO standard, it's also automatically recognized by all those regions. Using
It's
this.
reviewed by their standards committees and they decide how they're going to do it. That's part of a class I teach, is how to read the forward in a standard to see what parts of it are accepted by the different countries. And it's an easy thing to look at, but every country has their own standards group, and then you've got to check to see if it's been accepted there.
So you first must check if it's if your method of choice is allowed for the place where you want to send it to.
Yes.
Okay, thank you very much.
Where do you teach, Jan?
It's just an online class. I'm hoping I can do more somewhere along the line, but right now it's only an online class.
Well, you're welcome to put that in the on MDG Premium so folks can access it.
Well, we're just in the process of splitting it up because people didn't want to do 3 half-day sessions for 12 hours. So now I need to split it into 2-hour classes, and I haven't done that yet. So
Bruce, your camera's off.
There it is. Yeah, Kurt, I'd like to just hit a couple more technical questions before we get too far away from it. So this bag gets to the end user with its sterilant activity and the component that the end user actually wants. And if they have a great number of these bags to open, my question is, by opening these bags, will there be any exposure to those personnel that are opening these bags to take that product they want out of the bag?
None.
Okay, that's great. That one I was kind of stumbling on. And would this technology sterilize through another plastic bag? In other words, for example, an IV bag. Could this be an overwrap to an IV bag?
It
that
could.
Generally would get put in a terminal sterilizer normally, but to avoid that process of going into a terminal sterilizer, would this in
Yes.
fact? Okay.
Yeah. Sorry for answering before you were done. I apologize.
No, that's fine. That's a great answer.
yeah, I
Thank
find
you.
it good, useful for pre-filled syringes where we want to, you have a aseptically filled product inside of there that's going to be injected into someone, but you want to make sure the outside is sterile for the injection too. And that's always been difficult to get done.
I hear the words syringe and I think of our friend Dr. Carr. I wonder if when Kurt's over the FDA finish line, if his technology could be useful for her.
Oh yeah, definitely.
Well, be sure to, between us, let's be sure to have her watch this and reach out when it's appropriate.
What I've liked about it, you can stick it in there, and that bag, why he says the pinhole works, and you probably would still have a sterile product with a pinhole, is because the nitric oxide stops when it absorbs all the oxygen and kills everything. But there's still stuff left inside of that laminate layer so that if it gets exposed to oxygen, it starts working again and releasing. I don't know how long that release is, but that's something that is intriguing to me too for this technology.
How long it lasts depends on a few parameters. And it's tunable in the polymer. How you tune it, I have no idea, but Megan's taught me that much.
Kurt, given how engaged your fellow panelists have been today, each of them with questions and interactions, how would you wrap our call, and what would you say are the things that, if you remember nothing else from this call, what is the main point you'd like to make?
You no longer need sterilization facilities. That's one.
I immediately, like my hackles go up thinking about the competitive threat that is to an entire industry looking to flummox you at every turn. They would like to see you fail miserably.
Oh, yeah, and they mentioned that, by the way.
Tell me about that.
Well,
Just playfully at a trade show, or like, no, serious anti-competitive activity?
no, worse than that. I coach when I'm not doing this, and
What do you coach?
football in the fall and baseball in the spring. And the father of a guy that I coached walks into this restaurant that I was in Midland. And he comes over and taps me on the shoulder and he says, "Come here, I want to talk to you." And so I get up, I walk down the aisle with him. And he says, by the way, he works for DuPont, because with our system, you don't really need Tyvek anymore. And so he says, "Yock," because that's how they know me. "I heard that you're trying to take some of our business away." And I said, "Tom, look, there's something that you probably need to know. And we're not trying to take some of your business. We're, trying to take it all." And he didn't think that was very funny. And we aren't trying to take it all, but he's a guy that I've known for a few years anyway. And he was upset by the prospect that we might be changing the industry.
I would think.
Yeah, people in the industry, well, at least packaging engineers that don't work for DuPont, really want to get rid of Tyvek because they're expensive and they're the only ones out there that does what they do. And anything to get rid of Tyvek makes people happy.
Kurt, how are you aware of other companies working on similar technologies to yours?
Not that I know of.
Well, by similar, I mean other Tyvek hurting technologies, not just the way you do it, but this notion of letting your package sterilize your product.
I don't know of any.
I don't know of any either.
And since you do have a legal background, do you believe your intellectual property is adequately protected against someone else adopting some of this should you get clearance?
I spent a lot of money on it, so the answer is yes. And more than one law firm, because that really wasn't my area. So I had several firms look at it very carefully, and they tweaked a couple of things, but I think we're in good shape. We have had patent applications come back where we had 52 claims, and none of them were turned down.
So what
And
you're saying is I may have just had as a guest on our meager little show that's going to be a big fucking deal. I'm going to be like, yeah, I know Yock.
You just
Perhaps.
disparaged the rest of us, Joe.
Yeah, right.
I disparage the rest of you?
Everybody.
Implying that none of the rest of us are big deals.
I didn't say that. But you remind me, Kevin, that I know a couple of good therapists, and if you need to talk to anyone, I can.
I asked to talk to you just the other day and you turned me down.
That's not true. But actually, we're at the top of the hour, so if you're not busy, give me a call after this. All right. Kurt, Yock, thank you.
Ben, thank you for having me.
Yeah, no, I as a marketing strategist, I'm in the minority here. Yes, I'm an engaging host, but I know nothing of polymers and all of this stuff that's way over my head. But obviously our audience knows it quite well and found you very engaging. And apparently you've already lined up Andre as a future customer.
I'm happy to talk with anyone who would like to. It's sterilestate.com.
Okay, sterilestate.com. And of course, Jan, thank you for introducing us. You can go through Jan and she'll tell you what she really thinks of Yock. Next week, we have a guest going to talk to us about bringing a new contraceptive to market. In the weeks that follow, we'll have regulatory, CAPA, embedded software, and a talk about funding. So keep it tuned to this dial. And for Joe Hage and Joe Hage Enterprises, this has been Joe Hage. Thanks for watching, and we'll see you next week.
Thank
Bye,
you.
everyone.